Hi, so in this video today, let's talk about membrane transport. So we have a lot to talk about, from passive transport to secondary active transport. But before that, if you are seeing this video, make sure that you like, make sure that you share, and please subscribe. Well, to really understand the membrane transport and to go about all membrane transport mechanisms, it will be a good way if we went through the cell membrane, see its structure and see how it functions, and from there we'll be having an easier way of understanding the concept behind each and every mechanism. So if you look at my diagram here, you'll see that I've drawn a very good diagram of the cell membrane. You see these things that you're seeing here? These ones are the phospholipids, so which are made up of phosphates and sphingosines. And you can see that they are facing on the inner layer of the cell membrane and the outer layer of the cell membrane. And that is to mean that they're interacting with the fluid on the outer surface and the fluid on the inner surface. So by looking at this, you'll see that I've drawn other extensions from the heads of these phospholipids. Those ones are the fatty acid tails. So the membrane lipids consist of phospholipids, that is one, which are made up of phosphates and sphingosines, and the next thing, they are made up of fatty acid tails. And you can see that apart from the phospholipids heads, the fatty acid tails are looking away from the fluid from the inner layer and fluid from the outer layer. And that is to tell you that these fatty acid tails do not interact at all with the water. And so these fatty acids, they are hydrophobic. If they are hydrophobic, and looking at the phospholipids heads, which are looking at the inner layer and the outer layer, that is to tell you, for phospholipids, they love water, and so they are hydrophilic. That is a big difference. So fatty acids, apart from that, they are differentiated with two things. First of all, there are those that are saturated. I'll explain what that means. And there are those ones that are unsaturated. For saturated, it just means that they are made up of double hydrogen bonds. So these ones are made up of double hydrogen bonds. And for unsaturated, they are made up of a single hydrogen bond. It is as easier as that. So still at the membrane lipids, there are other structures. Do you see these structures that I've drawn inside these fatty acids with my pink marker? Those ones are called cholesterol. So with membrane lipids, there are three structures to look at. We have got phospholipids, we have got fatty acids, and we have got cholesterol. What cholesterol do is that they are involved in regulating the fluidity within the cell membrane. And what this basically means is that they allow the flow of the materials that are moving inside the cell and outside the cell. And so for cholesterol, when temperatures are high, guess what they do? They allow for these phospholipids to stay away from one another. And when the temperatures are low, they prevent these phospholipids from sticking closer to one another. So these cholesterol are involved in fluidity. So with that, we can clearly say that we've looked at membrane lipids. Let's now go ahead to look at membrane proteins. So if you go back to my diagram here, you'll see that I've drawn an extension here of this structure that is spanning the entire cell membrane, and it moves from the inner layer all the way up to the outer layer, and this is a membrane protein, but it is a different membrane protein because this one is an integral protein. So this one here, it is an integral protein. And another name for an integral protein, it is also called intrinsic protein. And these mostly are the ones which act as ion channels, because you can see that they occupy the entire cell membrane, allowing for substances. And with substances, I mean charged molecules or polar molecules to pass through the cell membrane inside and outside. So these integral proteins are made up of strong covalent bond. I'm sure you've ever heard that, is that they are made up of strong covalent bond. So coming down here, you'll see another structure right here, which, unlike this one only occupies one side of the cell membrane. So this one that I've drawn with my blue marker, this one will be the extrinsic protein, and the other name for the extrinsic protein, it is also called the peripheral protein. So this one is a peripheral protein. So if the integral protein is made up of a strong covalent bond, guess what? This one is made up of a weak bond. Well, so going ahead, there is another structure that we must talk about, and it is this structure that is coming off this peripheral protein. That structure there, this one, it is a carbohydrate chain, and that one is called a glycoprotein. So let's talk about the glycoprotein. So there are other carbohydrate chains that are coming off the head of the phospholipid. Those ones are called glycolipids. But for this one, because it is coming from off the peripheral protein, that one will be a glycoprotein. So what this structure does is that it is involved in a number of activities. And one is that it is involved in cell signaling. And what that means, signaling, is that it acts as a receptor where molecules and specific molecules will bind to. So one, it is act as a receptor, and that is through cell signaling. The other thing that it does is that it also gets involved in cell recognition, and with that it means that it can act as an antigen. So it is also used in cell recognition. Well, we've talked about the structure of the cell membrane and its functions, and with that we'll have an easier time to go about each membrane transport mechanisms, because this is exactly what you need to understand for you to get the whole concept of membrane transport. So let's go ahead and do that. We are going to start with simple diffusion. And one thing you need to learn about simple diffusion is that this process is a passive process. What does that mean? So this is a passive process, And what passive transport means is that simple diffusion is movement of materials, movement of ions, or movement of molecules from the region of high concentration to the region of low concentration, and this is happening at the cell membrane without requiring any energy. And the energy that we are talking about here is that no ATP that is needed. Well, if we go ahead, we've said that simple diffusion is movement of molecules. So, is it okay if I mention some of these molecules? One, these are respiratory gases: oxygen, CO2. What else? We have got lipid drugs. So we have got lipid drugs. Something else are hormones such as testosterone, estrogen. All these materials will go past the cell membrane either inside or outside the cell with ease, and they are moving from the region where they are highly concentrated, so from the high area of concentration to where? To the low area of concentration. And with that, because they are going downhill, they will be not needing any energy. That is it. So these molecules will move either from outside the cell to inside the cell, or from inside the cell to outside of the cell without needing any ATP. And that is what it means by passive process. And in simple diffusion, we have defined that oxygen, carbon dioxide, lipid soluble drugs, hormones like testosterone, estrogen, they walk through inside the cell or outside the cell with ease, from high area of concentration to the region where they are less concentrated. So with that, we can now talk about facilitated diffusion. So facilitated diffusion and simple diffusion, they differ in one way. You see, in facilitated diffusion, it is movement of materials, and these materials are also moving from the region of high concentration, and they are going where? To the region where they are less concentrated, so from high to low. So they are going downhill, and with that, it also means that they won't be needing no ATP. But where is the difference? So we've seen that in simple diffusion, these molecules, something that we did mention is that they are small. Something else is that they are non-polar. And with non-polar, what it means is that these molecules do not have charge on them. Examples are oxygen, CO2. They do not carry any form of charge within them. They are not negatively charged or they are not positively charged. And with that, they will move with ease through the entire cell membrane. But when it comes to facilitated diffusion, there is something different. These molecules that are walking through these channels here, they're gonna need a channel, and it is an ion channel, and they can require something that we call a carrier protein to move through the cell membrane. And why is that? In simple diffusion, we have seen that the molecules that are doing so are small, and that is to mean that the ions participating in facilitated diffusion are large molecules. And something else to sum up this discussion is that if the molecules going through simple diffusion process do not have charge on them, How about in facilitated diffusion? These molecules are charged either positively or negatively, and with that we can say that the molecules going through the facilitated diffusion are polar molecules. Examples are the electrolytes like potassium, sodium, protons, and all that. Well, let's talk about now the examples found within facilitated diffusion in order to get to understand each and how different they are. We'll start with osmosis. Osmosis is part or is an example of facilitated diffusion, and we say that osmosis is movement of water molecules from the region of high concentration going to the region where it is less concentrated. But we've said that in facilitated diffusion there is one thing that is needed, and it is a carrier protein. So a carrier protein that is used in carrying water from high region of concentration all the way to the region where it is less concentrated is called an aquaporins. Aquaporins. An aquaporins is an ion channel that is used in osmosis in carrying out this process. There's something else that I want to add on this process of osmosis. We say that osmosis is movement of water molecules from the region of high to the region of low concentration. But there's something else. Let's say we know that materials like salt, sugar, glucose all dissolve in water, right? And let's assume that outside the cell there is high concentration of something like glucose, and inside the cell there is less concentration of glucose, and here we have got water molecules. Where is water going to move to? So we say that osmosis is movement of water molecules from the region of low solute concentration. So water will be moving from the region where there is less concentration of glucose to where glucose is highly concentrated. So apart from water moving from the region of high to low, we say that osmosis is movement of water molecules from the region of low solute concentration to the region of high solute concentration. That is it. Well, from there, let's now discuss about the leaky channels. In leaky channels, we say that we know that potassium is less concentrated outside the cell, right? And potassium is in high amounts where? Inside the cell. So this is the inside of the cell. So we have got a lot of potassium inside here, and there is less amount of potassium outside the cell. At the same time, we know that sodium is in high amounts outside the cell, and sodium is in low amounts inside the cell. And for facilitated diffusion, the idea here is moving of molecules from the region of low concentration to the region where they are highly concentrated, but we are using an ion channel or a carrier protein. That is the only difference. So potassium will be moving from inside the cell to outside of the cell, while sodium will be moving from outside the cell to inside of the cell, down their concentration gradient. Leaky channels, what they are important for is something that we call the resting membrane potential, whereby in action potential more of potassium will leave the cell, where a lot of sodium will be moving inside the cell. making the inside of the cell more concentrated with sodium and creating something like an electrical difference. And when that happens, normally we know that potassium is high inside the cell, but because now it has left the cell, it will need a leaky channel for it to go back to the normal resting membrane potential, which is negative 75 millivolts. That is it. Ligand-gated ion channels. We say that ligand-gated ion channels, these are channels that open and close, but it is subject to the binding of specific molecules that we call ligand. And an example of a ligand is neurotransmitters. So what happens is that in this channel, they're involved in something that we call the NMJ, or the neuromuscular junction. And what happens at the neuromuscular junction, we are trying to create signaling, a signal which is moving from the neuron. It is walking through to the synaptic cleft, and it is moving to the muscle cell. So what happens is that we have got an electrical signal, then we have got a chemical signal, and again we have an electrical signal in the neuromuscular junction. So what really happens is that within these neurotransmitters, something like acetylcholine. You see what I've drawn here? That one is acetylcholine. And this chemical messenger, once it attaches itself to this channel will open up like that. And once it opens up, it allows for electrical signal to cross over from the neuron into the synaptic cleft and into the muscle cell, creating what? Creating muscle contraction. That is it. So, voltage-gated ion channels. So these channels are channels that open and close, and this is subject to the electrical changes that are happening within the cell membrane. So I will explain what that means. So we know that in resting membrane potential, we have got negative 25 millivolts, right? But something like sodium can hit this cell membrane, and once it enters inside the cell, it starts creating a sodium concentration inside the cell until it triggers this cell membrane or the neuron to move to what we call a threshold for an action potential to happen. So something like from negative 75 to negative 55 millivolts. And once this happens, what will happen next is the rise or the influx of the sodium channels inside the cell, something that we call depolarization. So what happens here is that sodium is high outside the cell, right? And sodium is in low amounts inside the cell. So these voltage-gated channels, because they are specific to specific ions like sodium or potassium or calcium, sodium will be moving from outside the cell where they are highly concentrated to inside of the cell where they are less concentrated, to create this high concentration of sodium inside the cell membrane, causing this change of resting membrane potential, creating a threshold of negative 55 millivolts, and creating what we call an action potential. Active transport now. So we say that passive transport is movement of molecules, and they are moving from the region where they are highly concentrated to the region of low concentration. In other words, this movement is happening downhill. And in passive transport, no ATP that is needed. So when we talk about active transport, we are talking about movement of molecules from the region where they are in low concentration, and they are moving where? They are moving to the region where there is high concentration. And so this is like they are doing this against the concentration gradient. And this against mean that if they are doing so against the concentration gradient, ATP will be needed. Isn't that right? Now, we have got primary active transport and secondary active transport. So in primary active transport, we say it is movement of molecules from the region of low concentration to the region where they are highly concentrated against the concentration gradient using ATP. But how? Directly. So let's find out how that happens by going through this primary active transport mechanism. We've got sodium potassium ATPase, we have got proton pumps, and we also have got calcium ATPase. Well, sodium potassium ATP pump is explained this way. One thing you need to know that these are potassium molecules and these are sodium molecules. And so something else is that sodium is highly concentrated outside the cell. So this is sodium, and sodium is highly concentrated outside the cell. And at the same time, potassium is in low amounts outside the cell. So this is our potassium. So what this sodium potassium ATP pump do is that they have got special enzymes on them, and these enzymes are called ATP pump or ATPase. So what these ATPases do is that they take something like ATP and they change them into ADP. Watch out here. So this ADP, what it does is that it changes the entire shape of this ATP pump. And by changing this shape, this is what happens. So look what happens. They change the shape of this entire ATP pump, and this is what happens. They take by changing the shape, they have allowed 3 sodium molecules to move outside of the cell by every 2 potassium molecules. This ADP changed the shape of this ATP pump by allowing, and by doing that, it takes 3 sodium molecules outside the cell for every 2 potassium molecules that are moving inside of the cell. And the idea here is, if sodium is high outside the cell and it is low inside the cell, it means that sodium will be moving from inside the cell to outside of the cell, right? And this is what happens here. for this ADP to change the shape of this sodium potassium ATP pump and allowing these sodium molecules to move from inside of the cell to outside of the cell against their concentration gradient, explaining how the sodium potassium ATP pump or the sodium potassium ATPases function. Now, proton pumps or hydrogen potassium pumps, but entirely these are proton pumps. So one thing you know is that we have got high amounts of protons inside the stomach lumen or inside of the stomach. High amounts of protons. But when it comes to where the protons are made, and that is at the parietal cells, there is less amount of this hydrochloric acid. And so what it means is that these protons will be moving from the region of less concentration, and that is from parietal cells, going to inside the stomach lumen against their concentration gradient and using ATP. Well, look here. There are patients who produce a lot of these protons. And by producing these protons, it means that they are at risk of developing diseases like peptic ulcer disease or gastroesophageal reflexes. And with that, it means that if they are producing tons and tons of protons, taking them to the stomach lumen, it means that this will increase the severity of this condition. And so there are drugs, and these drugs get called the proton pumps inhibitors or PPIs. So what these medications do is that they inhibit this process, and by inhibiting this process, it means that there will be a movement of these protons from the parietal cells into the stomach lumen, and that is to reduce the severity of these conditions like peptic ulcer disease or gastroesophageal reflexes. Well, lastly, are the primary active transport mechanism, which is calcium ATPase. So for calcium ATPases, what we are doing here is that we know that calcium is in high amounts outside the cell, and calcium is in low amounts inside the cell, right? So this is calcium. So calcium will be moving from the region where they are less concentrated to the region where they are highly concentrated. And this is important in where? During muscle relaxation. Because if we want the muscle to relax, will we be needing more of calcium inside the cell? No. And so what happens here is that these calcium molecules that are inside the cell, inside the muscle cell to be specific, will be taken from the muscle cell inside the sarcoplasmic reticulum, which is the cytoplasm of the muscle cell, where they are going to be stored. Because the idea here is taking lots of calcium inside the cytoplasm where they are going to be stored, because we want muscle relaxation for whichever patient. Secondary active transport. We saw that in secondary active transport, we are moving more than one molecule, and we are moving them either towards the same direction or we are moving them in opposite direction. Let's say that you have got molecule A and we have got molecule B. And these two molecules, both of them are outside the cell. And these two molecules are trying to move inside the cell. So this is inside. They are trying to move inside the cell. But molecule A has got ATP on it. And in secondary active transport, you must consider the sodium-potassium ATPases, because in secondary active transport, all mechanisms are dependent upon the sodium-potassium ATPases. And this is how. By taking 3 sodium molecules outside the cell and taking 2 potassium molecules inside the cell, we have created a concentration gradient of sodium outside the cell. So molecule A in this case is going to be sodium, and molecule B in this case can be either an amino acid or a glucose. And so that is to mean that if there is high concentration of sodium outside the cell, sodium won't be needing any ATP to move inside the cell. So it will be going down its concentration gradient with ease because there is no ATP that is needed. But for molecule B, it will need an ATP for it to carry or to go through that process, because it will be doing that against the concentration gradient. Glucose is in less amount outside the cell, and it is in high amount inside the cell. And so for it to move inside the cell, it will require ATP to move up against its concentration gradient. So in secondary active transport, we say that these molecules that are trying to move at the same direction, one is moving down the concentration gradient, while the other is going up against the concentration gradient, but it is taking advantage of the molecule that is moving down the concentration gradient for it to use or to utilize the ATP that is found within it. And so that takes us to our first mechanism of secondary active transport, which is the sodium glucose symporter. So in secondary active transport, if molecules are moving towards the same direction, they are called symport. And if they are doing or they are moving towards the opposite direction, they are called antiport. So sodium and glucose will be moving where? Towards the same direction. So look here. Sodium is in high amounts. So this is outside of the cell, and sodium is in high amounts. It is high. But inside the kidney tubules, there is low sodium molecules. So at the same time, glucose is in low amounts inside the kidney tubules. But glucose is high. This is glucose, where? Inside the kidney tubular cells. So these two molecules will be moving towards the same direction. Sodium is doing that down the concentration gradient, and sodium is moving up against the concentration gradient, wanting to take advantage of the sodium which is moving towards a similar direction so that it can utilize the ATP that is found within sodium, right? And with that, glucose will hang on the sodium for it to move from the kidney tubules inside the kidney tubular cells. Maybe if I could give you one more example, it will be an easier way of you getting the concept of the co-transport mechanism. Let's say we have got two persons here, person A and person B. And what these two gentlemen are trying to do is that they are trying to have fun by hanging on a seesaw. And one thing for sure is that person A is more weighty than person B. So let me draw person A using a different font here to imply that they carry more weight, and person B with a different font to imply that they are less weighty, that way. And so they are trying to have fun by sitting on the seesaw. And when person A sits on it and person B sits on it, obviously person A, because he is more weighty, he is going to carry person B up the ladder on the other side of the seesaw this way, right? So this person A, let's call him sodium, and person B is glucose. So person A has moved down the concentration gradient from here to here, right? While person B has taken the advantage of A moving down the concentration gradient for him to move up against the concentration gradient. It is as easy as that. So why is this important? So we have got patients, and these are diabetic patients, and we know that these diabetic patients, they have tons of glucose in the blood, right? And what we are trying to do here is that we are trying to reduce the amount of glucose in their bloodstream. And so we have got drugs, and these drugs get called the sodium glucose type 2 diabetes inhibitors, SGLT2 inhibitors. What these drugs do is that they inhibit the sodium glucose importer, and by doing that, sodium is not going to move down the concentration gradient from the kidney tubules to kidney tubular cells. So if sodium is not going to move, guess what? Glucose is also not going to move. And with that, sodium is not going to be reabsorbed into the kidney tubular cells. And so what happens with these patients is that they pee out a lot of glucose via their urine. Now, the NaKCl symporter, or the sodium potassium 2 chloride symporter. Well, what are we looking at here? This is in the area of the kidney, and specifically the powerhouse of the kidney, which is the nephron, and this is what we call the loop of Henle, or the LOH, and specifically the thick ascending limb of the loop of Henle, which is the ALOH. And so what is happening here is that this again is a symporter, or sodium potassium 2 chloride co-transport mechanism. So inside the cell we have got a lot of sodium, right? And you also have got a lot of chloride. And we know that where there is sodium, water gets pulled, right? So what happens here is that at the loop of Henle, and specifically at the ascending limb of the loop of Henle, there are drugs, and these are the loop diuretics. The loop diuretics work at a specific point of the nephron called the ascending limb of the loop of Henle, and what they do is that they cancel this process. They disallow sodium from being reabsorbed, and at the same time they disallow chloride from being reabsorbed back into the bloodstream. And if sodium and chloride are not going to be reabsorbed, water as well is not going to be reabsorbed, and so water get cancelled. And once this happens, is that these electrolytes will form something we can call a traffic jam at the ascending limb of the loop of Henle, which eventually is going to be thrown out of the body via what? Via the urine. So we talked about the secondary active transport, we talked about the symporter, which are the sodium glucose symporter, the NACACL symporter. Let's now finish up with the antiporter mechanism. So we've said that the core transport mechanism involves either an antiport or a symport. So symporter is when molecules are moving towards the same direction, and for antiporter, these are molecules that are moving against or towards the opposite direction. The first one here is sodium protons antiporter. So what are we looking at here? This is the area of the nephron called the collecting duct. And at the collecting duct, we know that there is a hormone. And this hormone gets called aldosterone. What aldosterone does is that it gets involved in reabsorption of sodium. And once sodium gets reabsorbed, water follows sodium. In sodium protons antiporter, what happens here is that sodium moves in while protons leave. So this is sodium while protons leave. And when there is less amounts of protons in the blood, guess what happens? There is metabolic alkalosis. So for patients who have got low amounts or low activities of aldosterone, so aldosterone activities are low, it means what? It means that sodium will leave, that is sodium, while protons will move in. These are protons. So when there is high amount of protons in the blood, what is that called? That is metabolic acidosis. The sodium-calcium exchanger. In sodium-calcium exchanger, this is an antiport. Or in other words, these two molecules are moving at the opposite direction. And we said that in every secondary active transport mechanism, it is dependent upon the sodium-potassium ATPases, whereby a concentration gradient of sodium outside the cell is created. And so, if there is high concentration of sodium outside the cell, sodium will do what? It will move inside the cell, and by moving inside the cell, calcium leaves the cell. So there are drugs, and these drugs are called the sodium potassium ATPases inhibitors, and these medications are utilized by heart failure patients. What happens is that these medications inhibit the sodium potassium ATP pump. And by inhibiting the sodium potassium ATP pump, it means that there is no concentration gradient of sodium outside the cell which is going to be created. And if that's the case, it means that sodium is not going to enter inside the cell. And if sodium cannot enter, it means what? It means that calcium is not going to leave the cell. So an example of this drug is a drug that is utilized by heart failure patients called digoxin. And what digoxin does, by inhibiting the sodium potassium ATPases, sodium is not going to enter the cell, calcium is not going to leave the cell. And calcium is very important, especially in reactivating or stimulating the muscle contraction, especially in heart failure patients, increasing the contraction. And by increasing the contraction of heart muscle cells, these patients have chances of surviving. That is it. Well, that is all for today. We've looked at membrane transport. We have gone through all the membrane transport mechanisms, from passive transport to active transport. And I know that this video was helpful. And if it was, make sure that you like, make sure that you share, and please subscribe.